So briefly, I want to touch on HVAC. I do still want to get to utilities before we have to hop off here. But Herman, would you mind briefly explaining for our audience the different considerations for HVAC when it comes to the different biosafety levels and modalities, as well as how manufacturers can reconcile and apply the diverse guidance across regulatory bodies from the FDA to the EU and CDC, et cetera, when designing their HVAC and key
Okay.
design principles around there?
Okay. A critical part of any containment in any type of facility, other than sanitization, is your HVAC system. Because the HVAC system is the only other active system that captures and eliminates any of your contaminants. And so it's an active containment system that uses air pressure. So that's the first thing. We use the HVAC system to do a number of things. We use it to protect the employees, to protect them from the what they maybe work with, a biological, an ADC or potent compound. We use it to protect the environment so it doesn't go out into the environment, the offices, and the land, and to protect the product from being reinfected. There's been situations where product has actually become reinfected from the original seed culture. BSL-1 is the first. It's generally not harmful organism to a healthy person. It's well characterized. It's minimum impact on the environment. So when you hear the word BSL-1 and you have to apply HVAC, you'd use your normal GMP gowning, Annex 1, and you can use a positive air pressure. You can use just basic good lab practices over GMP practices. And that's for organisms like E. coli, simple little guys like that. BSL-2 is the next level. We see a lot more of that now. And that's where BSL-2 could activate a moderate potential harm to people or the environment, where there's a therapy and a vaccine that exists. We have to contain that. So we typically, we focus a little bit more on recirculation, but we use HEPA in and HEPA out. So we have a continuous way of cleansing the environment to keep the minimization of the contaminants down. We also use a little bit better gowning and path control, whatever, on that. So HEPA in and HEPA out for BSL-2, and the same thing goes for BSL-3. BSL-3 is for more dangerous and exotic agents that can cause serious or lethal disease due to inhalation. And usually you have to do a lot of containment with using different types of mechanisms we'll talk about. The BSL-2 is things like seasonal flu. BSL-3 is like Q fever and botulinum. We don't want to go into the BSL-4 area. That's where like Ebola virus. So the concept with HVAC is to contain using air pressures. Now, when you go in and you have these from the BSL levels, you will then go into either the CDC or the European ECBC, and they will have, based upon organisms, the agents that they have, and they will actually list what's BSL-1, BSL-2, large scale, small scale, whatever. And from there, you'll be able to go in and identify what kind of containment you have. The BSL-1 means generally one layer of containment with air or with gowning. BSL-2 means 2 layers of containment with air and with gowning. BSL-3 is 3 layers, and 4 is, you better be careful about 4 anyway. So typically what we do is we use different types of systems. We use air sinks and we air bubbles to basically keep the air pressure in the area where an organism may be released through open vessel or through leakage. Okay, so we use those particular types of mechanisms to keep the containment. We understand the containment from first the CDC and the ECBC. That gives us our basic guidance of how to build. The FDA and the EU, when you're working with and getting the licensure from, will be in contact with you associated with the organisms and also with their counterparts in ECBC or CDC. In the United States, you also have to be careful about NEPA, which is the environmental folks that are interested in what happens to the environment, water systems, air systems, and so on, and a lot also with the typical EPA. Your transport of any kind of materials that may be infective. The NIAID provides guidelines on that. Your municipality and the countries also have a lot to say. So I could say the ECBC tells me that my flu vaccine is BSL-2 in the C train and BSL-1 during the delivery. Okay, but now you got to go and ask, what does Ireland tell me? What does Germany tell me? What does Switzerland tell me? Because they actually have their own different regulations, and they may decide in one thing or another. If a particular organism is non-indigenous to that area, for example, then if you look at the United States, you'll have to go to the municipality. So let's go ahead and build a facility in Durham, like Fred and I and Eric got involved with in Morrisville, North Carolina. You have a microbiologist that comes to visit you. They ask you questions like, what type of organism? What was the BSL level? Where did you get it from? How do you transport it from there to here? How do you contain it? What mechanisms do you use? Let me see the facility. Let me see the HVAC. So they are very, very interested in that. So in that, and they'll also want to take a stream of water stream. So the answer is, when you look at HVAC, you look at different levels of containment based upon the BSL level. And on the potent compound basis, you also have band A, B, C, D, E, F, and materials, which also tell you the level of containment, which is also physical as well as HVAC. Yeah. So you got to look at those guys, and then that will tell you where you got to go.